Hello everybody, this is Vinya. Today I would like to solve few questions and give you few insights about the concepts involved in it. Okay, let's go to the first question now. Let's read it out. So, the first question says that as a given below is a pedigree showing the inheritance of an autosomal dominant disorder. Okay, so we need to assign genotypes for the person 1, 2, and 3. Okay, now solving pedigrees during examinations is time consuming. So to avoid that, we need to take few hints from the question itself. Like it mentions here that it is autosomal dominant disorder. Autosomal, which means the disease is expressed equally in both males and in females. Okay, and it's a dominant disorder. That means that presence of a single dominant allele is enough for the disease to be expressed. Say polydactyly or presence of freckles. These are all autosomal dominant disorders. For these diseases, the presence of allele which is dominant in homozygous form or in heterozygous form is sufficient. Say, for example, the same capital M allele is the allele which is responsible for causing polydactyly. Now, for a person to show the disease, he should have genotype homozygous dominant, that is capital M and capital M, or heterozygous, where a single dominant allele is present. Now, if the person is unaffected or for the disease to not to be occurred, one and only genotype in case of autosomal dominant disorders is small m and small m. Only in this case the person remains free from the disease. Now let's assign genotypes for the unaffected people in the pedigree. So this female is unaffected, so it will be of genotype small m and small m. This male who is completely fine will have small m and small m. And this female in the progeny will also have homozygous recessive alleles. So now our person 3 has been assigned with the genotype. Okay. Now let's go to person 1. Here we can assign two possibilities, that is, one being homozygous dominant and second being heterozygous. Okay, now let's assume if this is the case. In this case, one allele from this parent, that is capital M, and another allele from this parent small m might have been received by this progeny, but that is not the case. The progeny is exclusively small m and small m, that is homozygous recessive and free from disease. So this is not the possibility of occurring with genotype capital M and capital M. Hence, we can eliminate the options A and option B because it is giving the person one's genotype as capital M and capital M. So we can eliminate these two options. Now let's go to the person 2. Again, the same way, the possibilities will be only two, that is homozygous dominant and heterozygous. Again, in the same way here, you have capital M and capital M and small m in the pairing conditions. Now, when these both are paired, all the progeny will be heterozygous. That means all of them will be affected. But we have a free of disease person in the number 3 position. Okay, so in this case also we can rule out the possibility of person 2 being homozygous dominant. Now we eliminate this option which is showing person 2 to be homozygous. Now we are left with option D and we are left with our answer 2, that is we have capital M small m as the genotype of person 1, which is holding true here. That is, when in this situation only the person can be affected and also the progeny will be clear from the disease. And in the same way, in the second cross also you will have heterozygous for this person too, and in the same way the progeny will be free from the disease in such a cross. Okay, so our option D is right, and in this way, without wasting any time, without drawing all the checkered board analysis, you can just come to the conclusion and get to the right answer. Hope this answered your question. Now let's go to the next question. So it states that which of the following two out of four statements are incorrect? Okay, so let's see which of the following two out of the four statements are incorrect. It's showing that it is incorrect. We need to focus on this. Okay, so let's see. Let's read out the statements first. The first transgenic buffalo, Rosie, produced milk which was human alpha lactalbumin enriched. The second statement states restriction enzymes are used in isolation of DNA from other macromolecules. C. The third statement is downstream processing is one of the steps of rDNA technology. And fourth being disarmed pathogen vectors are also used in the transfer of rDNA into the host. Now, let us see which of these four statements are incorrect, and let's get to the answers by the same process of elimination. Okay, now let's see about the first statement, which is saying that Rosie was the first transgenic buffalo. This is incorrect because Rosie was the first transgenic cow. Okay, so now we can rule out the options B and option C because it is not showing statement A to be incorrect. Okay, now let's go to the other options. We have A and C, A and B. Now let's see what is C. Down, Downstream processing is one of the steps of rDNA technology. Now, this is true. Downstream processing is the final step in our recombinant DNA technology. So, as C, statement C is right, our option A is not right answer for this question. Now, let's go and see what is option B. Okay, restriction enzymes are used in isolation of DNA from other macromolecules, which is clearly wrong. And this, our last option is anyways right. This option is wrong. So, restriction enzymes are not used in isolation, but they will cut the DNA instead of isolating. Okay, they are the enzymes which are used to cut the DNA at specific sequences. Now, if we use them for isolation, what will happen? Your DNA will be entirely chewed up and you will not be able to get the gene of your interest. So, our right answer would be option D, stating that statements A and B are incorrect. Okay, now we got the answer. Now, let me give you a few insights of about these options. So, let's go to the first statement, which is the first transgenic cow, Rosie. Okay, this was produced in 1997. Okay, it has produced 24% lactalbumin enriched milk. Okay, now this was special because the milk which Rosie produced was nutritionally more enriched than the other cows. So it made for the humans also to be easy for the consumption of the milk produced by Rosie. So now let's see how was it produced. First, a gene, say this is the gene encoding lactalbumin, was identified. Now, then, a gene construct was made with this gene, which includes its promoter and other sequences. And now, this construct is inserted into a cow. Now, what will they get? They will obtain transgenic cells. Okay, and these transgenic cells were fused with oocytes. Okay, how were these oocytes? These oocytes were such a way that they do not carry any chromosomes within them. Okay, so that only the transgenic expression would be dominant. Now they After the fusion, a zygote was formed and eventually an embryo was produced. This was implanted into another cow. Now we have to wait for its gestation to be completed so that a calf is produced, and this calf is our Rosie. Okay, the gene encoding lactalbumin is present in all the cells of Rosie, but it is expressed only in the mammary glands. Hence, the milk was enriched with lactalbumin, and only during the lactation time you could observe the transgenic expression. So, this is about the first option. Okay, now let's go to the option number 2. That is, restriction enzymes are used in cutting of DNA molecules from other macromolecules. These restriction enzymes are the enzymes which are isolated from archaebacteria or bacteria. Okay, they are very specific and they cut the DNA at specific locations which are termed as restriction enzymes. Sorry, restriction sites. Okay. Now, for example, this is your DNA and there's a gene located here which is of your interest. Now, these are the restriction sites. Your restriction enzyme will act on these sites and it will create a nick on both sides and you will get your gene of interest. Okay, and this can be further used in DNA cloning experiments or even in the transgenics, right? Now, let's see what is about C. C is telling us that downstream processing is one of the steps of our DNA technology, and yes, that is true. Downstream processing is the final step of our DNA technology. Now, when you are interested in getting a transgenic product, whether it may be a medicine or it can be a protein or it can be any product of or a therapeutic product, okay, you will get it with in combination with various other compounds, various other substances. So, to get the product of your interest, you need to eliminate all the unrequired ones. So, that is downstream processing, and that is done in 4 steps. Okay, so first one is you need to take out or remove all the insolubles which are present along with your product of interest. Now, this is done by general filtration or sedimentation processes. Now, second one, after that step, you will have to isolate your protein or isolate your product of interest. Okay, this is done by solvent extraction methods. Okay, this is done because your product of interest will be combined with various other substances which are similar to its physical and chemical properties. Now, you need to employ techniques which will separate your product from all the similar ones. So, solvent extraction could be one method which you could use to isolate the product of interest. Now, third one being purification. After isolation, also, your protein or your product is contaminated with various other substances. So, that is purified, okay, by using chromatography or crystallization techniques so that your completely 100% pure product is obtained. Now, finally, you are left with packaging. Okay, you need to package the product which you have obtained and go for the further sales, right? Now, let's go to the last statement which is giving us the information about vectors. So, what are those? Disarmed pathogen vectors are used in the transfer of rDNA into the host. So, what are vectors? Vectors are nothing but vehicles. They transport the gene of interest from one cell to another. Okay, now these plasmids or fast DNAs are used as efficient vectors usually. For example, let me tell you about TI plasmid. This is obtained from Agrobacterium tumefaciens. It is a circular vector. Okay, now this is actually causing a tumor in plant. Okay, Agrobacterium tumefaciens is a bacteria that is responsible for the occurrence of tumors in plants because its plasmid contains a location or a locus which will induce tumors in the plant. Say this is the locus. Now, in the same plasmid, you are inserting your gene of interest too. Now, our goal is to transfer this gene into another host without affecting the host with any tumors. So, what is need to be done? You have to eliminate this region and recircularize the vector and send it to the host so that an efficient transfer of this gene of interest can be done without harming the host. This is nothing but disarming. That is, you're eliminating the pathogenicity or the virulence of the vector, and you're efficiently transferring the genetic material into another host. So, these are all the insights which I wanted to give you. Hope these insights and answers have helped you. Thank you.